Hello everyone, we are the members of Group 17. This semester we have been researching autoimmune diseases, and the topic we chose to do our capstone project on is rheumatoid arthritis, or more commonly known as RA. In this lecture, we will go over some key terms about autoimmune diseases, the pathophysiology of RA, the results from our GWAS study, our one-page research summary, and any remaining questions and discussions about our topic. By the end of this presentation, you should be able to identify and explain our key takeaway points from our selected capstone topic. They include, what is RA and why is it an important autoimmune disease? How do our selected SNPs interact with the genetic processes of the disease? What are the treatment protocols for RA? And lastly, what effects does the environment have on the development of RA and its symptoms? On this slide, we have some key terms gathered from different online databases that define what exactly constitutes an autoimmune disease. I'm sure many of you have heard of specific autoimmune diseases before, as there are over 100 different ones out there, each affecting their own set of genes. From this information, an autoimmune disease can be defined generally as a chronic disease in which a person's immune system attacks their own healthy cells. So what exactly is rheumatoid arthritis? RA is an autoimmune disease characterized by widespread inflammation in 4 or more joints, also known as polyarthritis. This disease arises from inflammatory cells and markers attacking the synovial linings of the joint capsules, producing an excessive fluid buildup. It most commonly affects smaller joints first, like the fingers and toes, but eventually will move into larger joints, such as the wrists, ankles, knees, elbows, and hips. Usually the same joint on both sides of the body will be affected and express the same signs and symptoms. The increased swelling in the synovial linings can create an extra layer of tissue known as pannus, which will harden and contribute to the stiffness felt in joints. Over time, this can lead to severe cartilage and bone erosion and ultimately joint deformity. Some typical signs and symptoms that RA patients present with are joint pain and stiffness, chronic swelling, muscle loss, fatigue and weakness, and sometimes fever. This disease affects multiple body systems and can lead to high risks of secondary effects such as cardiovascular disease, metabolic syndrome, and osteoporosis. Generally, people who are diagnosed with RA are 2 times more likely to also be diagnosed with any of the above conditions.
Educating people on RA is important because it is a common autoimmune disease. More than 50 million Americans are affected by one or more autoimmune diseases, where 75% of the affected individuals are women, and 70% of people who are diagnosed with RA are women. We are likely to encounter individuals living with this ailment in our lifetime. Identifying the early warning signs and symptoms of RA could help us significantly enhance the quality of life of those close to us. But ignoring symptoms can cause further damage to joints, skin, eyes, brain, or heart. 1% of the U.S. adult population, or approximately 1.3 million people, are diagnosed with RA. It is important to note that RA increases the risk of a heart attack and stroke by 60% within 1 year after diagnosis. Here's our group's graphical abstract about RA. We've included in the middle a joint that on the left is not affected by RA and on the right is affected by RA. You can see how that there's bone erosion, cartilage degradation, and reduced joint space that can make one very uncomfortable while doing daily activities. We also included a skeleton that shows commonly affected areas that an individual might have RA. Also, we included 2 SNPs that are very important in the association of RA and genetics, and also some other SNPs that were found in our GWAS study. Women are 3 times more likely to be affected by RA than men, so it is important for them to go see their doctor and make sure any early warning signs are checked out. Also, one can increase their risk of having RA if they smoke, are elderly, obese, or they have genetics that could cause them to have RA. Some symptoms of RA are pain and aching, stiffness, tenderness, weight loss, fever, fatigue or tiredness, and weakness.
GWAS stands for a genome-wide association study that utilizes multiple sets of DNA in order to identify genetic variations associated with diseases. The results are then used to develop strategies to detect, treat, and prevent specific diseases. A GWAS can be used to determine the genetic risk factors of a disease across populations by identifying the associations between genes and diseases. This can lead to breakthroughs in medicine as the discoveries can help guide public health strategies in disease prevention and treatment. The GWAS meta-analysis we chose as a group identifies 7 new rheumatoid arthritis risk loci. Moving on to our research study, we analyzed and expanded upon the GWAS meta-analysis. The problem we chose was to identify the new associated genetic risk factors of rheumatoid arthritis in order to develop prevention and treatment strategies for this disease and other similar autoimmune diseases. In the graphical abstract to the right, you can see the SNPs that are affected and involved in RA and the affected areas of the body due to the disease. RA causes pain in the joints, and a diagram of the healthy and affected joint is shown. Women are 3 times more likely to develop the disease compared to men, and this risk can be increased due to personal lifestyle choices such as smoking or genetics. Some of the symptoms associated with rheumatoid arthritis include pain, stiffness of the joints, and fatigue. Moving back to the GWAS, we can see that the research consisted of a meta-analysis of over 5,000 RA cases who tested positive for the autoantibodies. The pie chart titled "The Number of Cases Reported for the Meta-Analysis of GWAS Results" shows where the origin and distribution of the case collections. The GWAS used logistic regression analyses to remove outliers, and it combined the results of the 6 meta-analysis collections. The results of the study indicated that there are 7 important SNPs associated with the risk alleles. The graph titled RA risk alleles and their strengths of association shows the 7 SNP genes and how well they correlated with the developmental risk of RA. These 7 genes were determined to have functions within the immune system, which confirms the presence of RA as an autoimmune disease. The limitations of this study are that it only takes into account populations of European descent. This can be seen in the pie graph as it only includes collections from North America and Europe. The result also fails to consider environmental and lifestyle factors which can influence the risk of RA. In conclusion, the total number of confirmed RA risk loci is 31 among populations of European ancestry. Understanding the specific SNPs and markers that are associated with rheumatoid arthritis is important for future testing and advances in developing new preventative treatments.
In this genome-wide association study, an array of cutting-edge technologies and analytical methods were utilized, each contributing significantly to the depth and accuracy of the genetic analysis. The study employed the Affymetrix GeneChip 6.0 platform for analyzing genetic variations in the BRAS collection, and the Affymetrix 500K platform for the WTCCC collection, ensuring high-quality genotyping. Furthermore, the Sequenom iPlex platform was a key tool for genotyping in various collections, including Canadian and Dutch samples. This platform was notable for its ability to analyze multiple SNPs simultaneously, enhancing the efficiency of the study. Kinetic PCR, used for genotyping the GCI and LUMC samples, allow the DNA to be precisely quantified in real time, a crucial aspect of accurate genotyping. In addition, the BioTro OpenArray platform enabled high-throughput genotyping, an essential feature for handling the vast amount of data in the NHS samples. Lastly, the use of PCA, Impute Software, Eigenstrat, and Plink provided a robust framework for data analysis. These tools were indispensable for addressing population stratification, imputing genotypes, and ensuring rigorous data processing and quality control. Each of these technologies and methods played a pivotal role in the success of this GWAS, allowing them to analyze vast genetic data with precision and reliability. Here are the most noteworthy figures from the study. On the left is Table 4, which provides a comprehensive view of the SNPs associated with RA. It details their genomic location, associated genes, allele information, and statistical significance from different analyses. The table is effective in presenting detailed data, especially when comparing multiple variables or conditions. It provides a thorough and structured view of the SNPs, their genomic context, and their association strength with RA. While this is useful, a visual representation which more clearly displays the strength of association, like Figure 2 on the right, can help identify the most significant SNPs at a glance. This figure also emphasizes the overlap between RA and other autoimmune diseases. It contains 18 SNPs, 5 of which were validated as RA risk alleles in the study, and these have dots in the innermost circle. 6 more which have suggestive associations, and 6 which had no evidence. On the outside of this circle, other autoimmune diseases like celiac, MS, and psoriasis are included, as many of these particular SNPs have also been proven to be risk factors for these conditions. This figure is significant because it condenses the emerging understanding of the overlap in the genetic bases of autoimmune diseases and presents it in one neat graphic.
After learning about the GWAS, we were left with a few remaining questions. For example, how did the GWAS results apply to the non-European populations? Our solution was to do a cross-population comparative study to discover any specific risk factors. Another question we had was, how do these new 7 risk factors relate to those previously known? Our solution was to compare the GWAS for multiple autoimmune conditions to further explore shared genetic markers. The last question was, what was the role of environmental factors in developing these new alleles? If we look at longitudinal cohort studies, we can observe interactions between the genetics and environment, and we could find these answers. Here are some other topics to discuss when looking at our GWAS. Epigenetics. Lifestyle and environment factors could modify the regulation of these genes. Epigenetics changes can be reversible. A deeper examination of the influence of epigenetic markers on these genetic variants could lead to breakthroughs in treatment. Are there any LSSI considerations? Concerns of genetic discrimination arise in the areas of health insurance and job prospects, for example. These findings necessitate the creation of policies that protect individuals' genetic privacy. What about genetic technology? This study's use of numerous advanced genotyping and bioinformatics platforms shows the necessity of continued advancements in genomic technology. These technologies are additionally useful for diagnostic testing, drug development, and reducing healthcare costs. Making genomic technologies more accessible and cost-effective will transform patient care from reactive to proactive. Thank you for listening, and we hope that you've enjoyed our presentation on rheumatoid arthritis.